Researchers have developed a technique to simultaneously measure electrical signals from 128 brain regions in awake mice. They then used this information to measure what happened to neurons when mice were given hallucinogenic drugs. The results showed unexpected and simultaneous synchronization between neurons from several regions of the brain. The idea that electrical oscillations in the brain could be used to teach us about our experiences was conceived several years ago. Halge and team are studying mice with Parkinson's disease who have problems with involuntary movements. Researchers found an 80 Hz sound, an oscillation or wave in an electric field, in the brains of mice with Parkinson's disease. It turns out that waves are closely related to involuntary movements. A Polish researcher observed similar waves after giving rats an anesthetic ketamine. Ketamine was administered at low doses to keep rats awake, and equivalent doses in humans caused a hallucinogenic experience. The waves they saw were in more cognitive regions of the brain than in the Parkinson's disease mice and at a higher frequency, but it still made us wonder if there was any connection between the two phenomena. Perhaps excessive brain waves in motor regions of the brain cause motor symptoms, while excessive brain waves in cognitive regions do not, says Per Halge, a neurophysiology researcher at Lund University. Cause cognitive symptoms. Per Halge's research team has developed a method that uses electrodes to simultaneously measure the oscillations of 128 different brain regions in awake mice. The electrical waves are caused by the cumulative activity of thousands of neurons, but researchers were also able to isolate the signals from individual neurons. For some of these areas, this is the first time anyone has succeeded in showing how individual neurons are affected by LSD in a waking animal. When we gave mice a virtual anesthetic drug, it was the first time anyone had succeeded in showing how individual neurons are affected by LSD. Sensations of LSD and ketamine, the waves were clearly recorded. Collective wave pattern. Although ketamine and LSD affect different receptors in the brain, they have completely different pathways in the nervous system. They result in similar waveforms even though the signals from the individual cells are different. Together. When the mice were given LSD, the researchers found that their neurons were inhibited, they signaled less, in all parts of the brain. Ketamine seems to have a similar effect on large neurons, pyramidal cells, whose expression is inhibited, while visceral neurons, which are smaller neurons, are only collected localized in tissues, increasing their signal.